Good morning, everyone. So we are going to go ahead and get started. Welcome to our first session, the ecological effects of deposition and climate change. And our first speaker is Emmy Felker-Quinn. So the floor is yours, Emmy.
Good morning, everyone. I'm Emmy Felker-Quinn of the Air Resources Division of the National Park Service. And I'm going to be talking to you today about ozone effects on tree growth and survival. I'd like to start with my standard disclaimer that the views are my own and those of my co-authors and not those of the National Park Service or the EPA. So ozone. We have decades of research showing that ozone affects plant growth, plant mortality, plant senescence. Today, when we talk about plant responses to ozone, we typically use the W126 metric, which is mapped here on the left. It's a 3-month sum of daylight ozone hours weighted so that higher ozone concentrations contribute more to the metric. I'd like to point out that what you can see for 2021 here is that W126 values are really high in the American West. When we talk about W126, we don't have many dose-response relationships. We're relying primarily on tree seedling responses from tree seedlings grown in open-top chambers. Species-specific responses from those studies done by EPA were just published last year. The figure is here on the right, and it really nicely demonstrates the diversity of responses of different tree species to ozone levels. Not just that some species are more or less sensitive, but that some species are very tolerant of low and moderate levels and then have a sharp decline when a particular ozone threshold is reached. This is particularly urgent because climate change will increase ground level ozone across the US. This figure on the left is a figure for the National Climate Assessment. In the lower scenario, where we have moderate emissions and moderate temperature increases, we'll see increases in summertime ozone. If emissions and temperatures increase at a higher rate, we'll see higher concentrations of ozone across the country. It's particularly urgent because, as we can see from this picture from Colorado, oil and gas and urban development are both continuing in the American West, where W126 concentrations are already high. If you've been around NADP for a while, you might be asking yourself this question right now: why ozone? And the answer is really that what we'd like to do for ozone, what we have already done for nitrogen and sulfur. So the Critical Loads of Atmospheric Deposition Committee has established dose-response relationships for plant species. So for example, on the top panel, we have the effects of nitrogen deposition on black cherry. Initially, there is a positive growth response and then a decline after we hit a particular level, our critical load level. And also acidifying effects on plants like the black walnut in the lower panel here, which responds to sulfur deposition with a decline just as sulfur deposition increases. And so we've used total deposition to really establish these relationships of species-specific responses of growth, survival, and abundance to deposition across the US. The figures from the previous slide and the figures shown here are from a Horn et al. 2018 paper, which published nitrogen and sulfur deposition relationships for tree growth and tree survival from 94 tree species broadly distributed across the US. And so what this talk will kind of attempt to answer is, can we use the same methods that have successfully established nitrogen and sulfur critical loads to establish ozone critical levels? Loads for deposition, levels for air concentration. If you're not familiar with the Horn et al. Paper, it used the Forest Service Forest Inventory and Analysis tree surveys conducted between 2000 and 2016. The initial analysis used species that were widely distributed that had at least 2,000 trees per species. And for each species, there were maximum likelihood models that included tree size, competition among individuals, climate data as mean annual temperature and mean annual precipitation, and then total nitrogen and total sulfur deposition. And so the figures on the right are kind of the deposition relationships that were explored by these models. Models were compared with AIC scores, and the winning model had the lowest AIC score and was the most parsimonious. So we are reanalyzing that Forest Service data. We're expanding the number of trees, species, by relaxing our requirement for how many trees are included to 500 trees per species. And we're adding ozone, so we're testing again for nitrogen and sulfur as we did before, but also adding ozone. The ozone data included in these models is an interpolation from ozone monitors of the highest annual 3-month W126 value. And these are done annually, and each tree is associated with the W126 values that occurred between its measurement points in the surveys. In addition to decreasing or threshold-shaped curves, we're also testing a tabletop relationship for sulfur and ozone. We're hoping with this relationship of ozone to growth or survival to really capture the fact that many species appear to be tolerant up to a point and then can have a really sharp decline. So we call that sharp decline point the peak growth or survival, kind of the tabletop edge. And actually, when we're looking in these models for tabletop or for straight decline models, tabletop wins every time. So 85 tree species had a winning growth relationship that included a tabletop relationship of ozone to growth, and 38 species had a winning survival relationship that included ozone in the model. Again, we're borrowing from CLAD as we move forward in evaluating these models. CLAD Working Group 2 has developed uncertainty categories, kind of similar to the weighted deposition uncertainty metric that TDEP has. And this allows us to compare our certainty in different critical loads for nitrogen and sulfur. So we look at variance inflation factor and correlation to give us a sense of how our explanatory variables relate to each other. And then we look at a pseudo R-squared just to see how well the model is fitting the underlying variation in the data. And so as we do this with the ozone models, what we're going to do is restrict consideration of ozone critical levels to models where all scores are at least 3. So we know that ozone, ground level ozone forms on hot, dry days where there's a lot of NOx. Remember that there's ozone in these models, but there's also precipitation, temperature, and nitrogen deposition. We're very concerned that we want to be able to say that these models are really about ozone and not one of the other factors that might correlate with ozone. So one of our results is this model of sweet gum growth and W126. We're showing the relationship here just over the range of W126 values of 3.7 to 28.4, which is where sweet gum grows in FIA plots across the US. Peak growth is actually fairly low in that range at 3.9 ppm hours. And if we want to use the 5% growth increase that EPA's CASAC has recommended as a critical level, it's at 5.2 ppm hours along this curve. We have fairly high certainty. Our most uncertain characteristic here is the correlation of ozone with sulfur, which is still within our kind of limits of what we're willing to consider. By comparison, that Lee et al. Paper that looked at tree seedlings established a critical level for sweet gum seedlings of 25 ppm hours. So compare that to 3.9 and 5.2 for mature sweet gum. We can also look at one of our models of a tree that has low certainty, and and that's ponderosa pine. It's one of the most abundant species in the West. It grows over a wide range of ozone W126, but there's a very strong correlation between ozone and nitrogen when you look at where these plots of ponderosa pine are. And so although we could pull out these critical levels for mature ponderosa pine from this analysis, we are instead going to continue to use to describe risk to ponderosa pine the tree seedling critical level from Lee et al., which is 6 to 9.6. So moving forward with model selection, we intend to extract quantitative relationships for species with high certainty scores. And so, excuse me, of significant models that included ozone, that's about half. So only 47 species for growth and 23 species for survival. Well, it's only about half the models generated. Compare it to the current information that we have for ozone, which is 16 critical levels for tree seedlings just for growth. So this is still a significant addition to what we know about ozone effects and quantitative relationships. We're also going to develop additional criteria as we go through these models related to model plausibility. This is the relationship of subalpine fir to survival. This is what my statistical professor called a facial trauma test, which is it hits you right in the face. This is not a significant model. Like, the model and parameter certainty scores pass our selection criteria, but really, when you take the survival rates over the range in which subalpine fir actually occurs and calculate it out to an absurd 9 decimal points, it really does not vary in survival at all over the range in this model. And so we're already starting to think about how we're going to be using these models. So when we have quantitative relationships with high certainty scores, we'll be able to use critical level language. So mature sweet gum has an ozone critical level of 3.9 ppm hours to protect peak growth, and it has a critical level of 5.2 ppm hours to prevent significant annual growth loss. For the subalpine fir flat line that I just showed you, where we're concerned that the model doesn't actually show us something biologically significant, we'll probably use language like this. We're fairly certain that survival of mature subalpine fir has no relationship to ozone W126. And then for the species that have significant models but lower certainty scores, we intend to qualitatively identify those species as ozone sensitive. So that might be something like ozone W126 reductions might benefit mature ponderosa pine growth rates, but we're not attaching a number to that. We're probably going to continue to use the tree seedling data there. And so with that, I want to acknowledge Sonoma Technology, who ran the ozone spatial interpolations, CLAD Working Group 2, who has spent years wrestling with certainty and uncertainty and kind of how those bins should be set. And you know, this is a reanalysis, and it's big. Lynn DePardo is leading the nitrogen and sulfur critical load analysis that comes out of this same model rerun. And with that, I'm happy to take questions and comments, or, you know, email me.
So are the values that are coming out of this initial analysis similar to previously measured ones like in open top chambers, et cetera?
That's one of the interesting things, is that from our first look, the values for the mature trees are actually much lower than they are from the tree seedling studies that are done in the open top chambers. Yeah, so 3.9. I mean, the critical level for the tree seedling is actually set for 5% growth, so it's directly comparable to the 5.2 for 5% growth decline in mature species.
When I see values that are so low for ozone, it's a bit concerning. Forest Service had a big study about a decade ago where we went to the inventory sites across all forested lands that are part of the forest inventory analysis that we're required to do by Congress to track the state trends and health of the nation's forests. And they had an ozone indicator which involved looking at symptoms in plants. And it found a few places where symptoms were detected, but actually there were quite a few areas where they didn't detect anything. So I'm wondering whether we should look back on that study to see if there could be any type of ground correlation, like where you could track levels and look for symptoms on the ground, or if there are no symptoms, why not? But anyway, it would be nice to do some ground truthing of that, and there might already be data that could help with that.
Yeah, I'm glad you brought up foliar injury, because that's been a big push with some of this monitoring, is to look for distinctive ozone injury symptoms on leaves of sensitive species. I would say that like we've looked for it at Rocky Mountain National Park and found it even in years where W126 has been relatively low. Cumberland Piedmont has looked at injury, and they see it, and it doesn't have a real good relationship of what W126. EPA did a big analysis of the foliar injury data from the Forest Service for the, what was it, the most recent ozone PA. And one thing that they have really looked at or asserted in their documents is that foliar injury and effects on growth and survival don't correlate.
Emmy, just following up on Mike's question, do you think that the response at lower levels for mature trees has to do with the cumulative exposure? Because obviously those open top experiments are kind of limited in duration. Is that part of what's going on?
I mean, both the seedlings and the mature trees are the 3-month exposure, and so I think that the metric is comparable between the studies. Yeah, practically speaking, even 5.2 is really low compared to, a species that goes up to about 30 ppm hours.
But the FIA analysis is, that's just over the long term, right?
Right.
So those trees could have been exposed to that ozone for decades, or anyway, years.
Mm-hmm.
Yeah, I guess just in relation to the chamber studies, that was based on the loss in biomass, correct? And so was the foliar injury also observed at these same critical thresholds or critical levels?
In the chambers?
Yeah, or was the loss of biomass observed without seeing foliar injury?
I think Chris Novak is going to answer this one, because I think he's looking at the foliar injury stuff now.
Yeah, so the, in those same studies, there were foliar injury analyses done, and we are currently trying to sort through that because it was a complex series of studies that were done in a couple different locations, under different PIs, and they tried to have a common set of things they looked at as a baseline. So we are currently trying to dig through those files and see what we can bring out for foliar injury, and then it would be a similar type of presentation as the lead paper, but it wouldn't obviously be growth. But in my experience with foliar injury and growth, you don't always have a strong connection between foliar injury and a growth reduction for every species, but there are some species where you do see that, and my own research in with European species that, there was strong presence of foliar injury, strong reduction in growth, strong reduction in gas exchange and photosynthesis. And then other species, it wasn't quite as, as coupled. So I think it's a species-specific type of response, but, in almost all cases, the presentation of foliar injury is an indicator of there being a phytotoxic response to ozone. So that's the least that we can say about it. But having always a strong quantitative relationship is challenging. So.
Okay, great. So our next speaker is Rob Smith, but Rob, it looks like we may not have your presentation. Oh, he's on.
Hey there.
Thank you. All right, thanks, Rob. The floor is yours.
Okay, thank you. I will share screen here in a minute. Is there a way to mute audio that comes back to me? Or can mute my own and ask for a visual if anybody needs feedback?
I think we have everything muted on our end, so let us know
Okay.
if we're problematic.
Got it, thanks. I will mute myself on my side. Okay, so I'm hoping to cover global change signal in lichens. Give me just a moment to change my audio. I apologize. So global change signal in lichens, and what I'm hoping to cover is global change from the perspective of climate and air quality and that interaction. So when we look at lichen communities, let me see here if I get my share. My apologies here. So I've often asked this question, what is the fate of future forests? What is the impacts of changes to climate and air quality regimes and also disturbance regimes on vegetation? So we have an idea about where future forests are headed. And we take focus on bioindicator lichens. So epiphytic lichens, those that grow on trees, I would consider the world's best three pretty much universally distributed solar-powered climate sensor network and air quality sensor network, if we are able to read their signal. So basically, we'll look at how to read that signal. One of the things we'd like to do here, though, is think about what we're talking about. So just for a brief review, a lichen is a fungal and photobiont partnership. If you take a cross-section of a lichen, the vast majority of that body is a fungus with some green algal partner or cyanobacterial partner, right? So we have a little bit of plant characteristic and a little bit of fungal characteristic. That's important to the basic biology because they have an atmospheric lifestyle. They're basically depending on any nutrition or any moisture that comes from the air exclusively. So we're decoupled from soil processes. Those are desirable characteristics for biomonitoring atmospheric conditions. Lichens are also used extensively throughout federal agency activities. So multiple agencies across multiple applications for multiple purposes to fit certain laws, regulations, and policies. So without exhaustively going through the list, we can say that lichens, especially lichen community monitoring, is extensively used in agency activities. So the goal here, if we're going to detect climate change signal, air quality signal, other kinds of changes in these global change drivers, we want to look at that across scales, from small local scales to nationwide and across multiple time scales. If we look at a single species at a single site under a range of potential future climates, as we've done in work with the Department of Energy in northern Minnesota. So the Spruce Project is a whole ecosystem warming project, a whole canopy warming project. And we have installed lichen transplants, the long story short of which is that individuals here at the bottom left of the screen, healthy green chlorophyllc individuals, as you apply heating, they become bleached out, less photosynthetically active, and they lose biomass. They're wasting away. That tells us there's definitely a climate response. Even after just 1 year of warming, we see a drastic change where as we add warming from left to right here on the gradient, we see drops in biomass. That is an average accumulation of maybe like 10% per year of mass growth in ambient conditions declines to where we're actually losing mass, we're wasting away. With some partners at University of Minnesota doing the ecophysiological measurements here, that's effectively due to the inability to maintain a positive carbon balance as warming is applied. So as you grow beyond ambient conditions, the lichens can no longer maintain positive carbon balance because their photobionts are dying out. That's evident just in that eyeball test where the green color, the healthy green color, fades away. So if that's what's happening for single individuals, how does that scale up to communities across large scales? So nationwide, we're lucky to have the FIA Forest Health, Forest Inventory and Analysis / Forest Health Monitoring / Air Resource Management Program data set. So that's systematic measurements nationwide of the entire lichen community that grows on trees and shrubs above ground. So we have nationally consistent monitoring and a complete species inventory at each of these sites. Given that data set, we can model where does one single species occur, for example, in this left-hand panel. If we take this species and say, well, that's where it occurs geographically, but with respect to its climate envelope modeled in the center here, we can also identify populations that are at the extremes of its tolerance, at the extremes of that climate envelope, and then map those back into space and say that those populations are considered quote unquote vulnerable. If I look at not just that single species, but what would be the percentage of species at a given site that is vulnerable, the percentage of species that is close to its climate niche, we find that that measure of thermal vulnerability ranges, but it often is most high where we also see impacts from poor air quality. So that's saying that we can't just measure the climate response without accounting for air quality and vice versa. We know that this is most commonly the more southerly low elevation sites where climate vulnerability is high, but that's also where we have air quality impacts. So given that, that's the predicted future vulnerability to climate change, but what has actually happened over a 2-decade span in one region? So with an intensification of measurement at 820 permanent plots in the Cascade and Coast Range of the Pacific Northwest, if you take a set of plots that have been sampled in the mid-90s versus those same plots measured again 20 years later, what we find just purely physically, those sites have warmed, right? So the color scale here, hotter colors equal hotter warming. Cooler colors indicate that that site cooled over time. Most of those sites did experience actual climate warming. So the question becomes, what happened to the lichen communities at those same sites? For single species, we find this pattern where the species that were already specialists, in other words, range restricted or habitat restricted, which are also a lot of nitrogen fixers that are important to ecosystem function, were the ones that turned out to be losers. So they had a higher probability of being lost over time, and at sites where they did not occur, they had a low probability of appearing later, right? So they were losers. The winners tended to be those that were already quite widespread and generalists. So this is sort of like a rich get richer and poor get poorer situation. Was there any change in community metrics at those sites? So there was no change in richness related metrics, but there certainly was in composition related metrics. So what that means is even though the number of species on average could remain constant, the identities of those species were turning over concurrent with the warming that was observed at these sites. That's mirrored when we look at the physical side of warming, so the actual climate warming experienced in this left-hand panel, compared to a composite measure of community composition. We see that the change in those lichen community compositions relatively mirrors the climate changes experienced. So to boil that down, lichens are responding at some time scale over 20 years to climate changes experienced. That's good. That tells us that we're measuring a sensitive bioindicator, something that's actually responding. When we look at relative importance of variables, I've been saying we can't ignore air quality variables. When we analyze those side by side, we find that climate warming, that's change in mean annual air temperature here, is the relatively more important variable in explaining community compositional change, relative to things like change in, this is climatic moisture deficit, so a drying or a moisture variable, as well as changes in nitrogen or sulfur deposition. So this is a typo here. So climate warming as an effect on lichen communities far outpaces other atmospheric drivers. So can we explicitly model, not implicitly, but explicitly model the climate air quality interaction? The reason we have to do that, as I've mentioned, is that stress tolerance responses in lichens are correlated. That is, the sets of species that you would see turn over as you walked from a relatively low pollution or a clean site along a pollution gradient to a relatively polluted site or a high deposition site would be the same turnover in community composition that you would expect if you started at the cooler, moist end of a climate gradient and went towards a hotter, dry side. So is this a liability or is this an asset? I hope to show how it could be an asset. So for this, the thing we want to measure, the lichen response we want to measure, is something called an air score. So this is, if I know for a single species what its tolerance is relative to deposition, in other words, its central tendency. And if I go to any given site and collect multiple species tolerances and take the average of that, that gives me something called an air score, right? So site 1 here, the collection of species have fairly low tolerances, so it would have a fairly low air score. Site 2 is kind of in the middle of the gradient, and on average, site 3 is relatively high. So this air score tells us something about the deposition as seen or as experienced by those lichen communities. And when we map those onto the landscape, we find relatively good correspondence between those air scores and the actual deposition experienced. You may also be thinking about how this overlays onto a climate map, because a lot of the low air score sites are in relatively cool or moist areas, and vice versa for the high air scores. So we can explicitly model this interaction with nonlinear regression, modeling the response of the air score, which when relatively low is like desirable set of species that is high ecosystem function, a lot of those nitrogen fixers, and then transition away from those to the more weedy pollution tolerant species or heat tolerant species, as represented by that high air score. And when we incorporate measures, climate measures such as maximum August temperature, in conjunction with the nitrogen deposition gradient, that's when we can see that interaction. So to view this in a different way, imagine that we're looking at the climate variable and the deposition variable in 2 dimensions, right? So this is that same regression I just showed you as a response surface rather than lines. Air scores increase with both deposition and mean annual air temperatures, or maximum August temperature, right? So low, you can think of this as a contour surface, just like a mountaintop, where the top of the mountain represents higher air scores, which represent community compositions dominated by pollution tolerant, heat tolerant species. And vice versa, down here in the valley would be these clean, cool species, that are characteristic of high ecosystem function, clean sites. So knowing this information, how could this help us with scenario planning? Imagine we know present day, here's my starting position. I have a site or a region of interest that has so many units of nitrogen deposition and currently is sitting at a certain climate regime. What would happen if we held deposition constant but allowed expected future climate warming to proceed? So you're basically walking along this contour surface, walking along this mountain for a do-nothing strategy. That is, keep deposition the same, but let greenhouse gas emissions and all the other things that might drive climate continue to occur. And with that, we see that we would hypothetically have, on average, a higher air score. We'd be climbing that mountainside hill. What would it take to keep a constant air score? So we can kind of invert that question and say, given this starting point, if I wanted to maintain a constant contour on this mountain, I would, and not interfere with climate, I would have to have a reduction in these non-greenhouse gases that drive nitrogen deposition, right, of a certain amount. So in a sense, this can be turned around to say that amount of deposition reduction would have a similar effect on lichen communities as offsetting the effects of the climate change expected, right? So it's a mitigation. If we can reduce that deposition by that amount, it may mitigate harmful effects on the lichen communities. Likewise, what would happen if we both reduce the non-greenhouse gases that drive nitrogen deposition and the greenhouse gases that drive the climate warming? We could actually see an improvement in the air scores where we come down a contour towards the set of clean, cool, desirable species. So it's just a way of using this surface to tell us what would happen under different hypothetical scenarios. So basically, lichens, in terms of community compositions, both climate and air quality have to be incorporated together in interaction, and that can help us with certain scenario planning. My co-author is listed here as a contributor, but she's a co-author, that's Linda Geiser, but I'd like to thank the following folks for their participation, feedback, and comments. So for listening.
Any questions for Rob? Oh.
Hi, Rob. Great talk. You focused on the Pacific Northwest and nitrogen. Do you think there's a similar story about sulfur in the Appalachians, or do you think that nitrogen really drives the lichen air scores there too?
I would suspect it would be both. So there's no particular reason why I picked on nitrogen other than it simplified the story. But they're certainly both occurring together. It's not something, especially with, we don't have adequate resampled plots there, but that would be helpful to kind of determine that response.
Check and see if there's any questions online. Okay, we do have 1 or 2 more minutes.
So Rob, how much variability is there in those contours? Are they regionally similar, or is it each site is kind of specific?
So though I showed you one example regression that is one of many that was part of a critical loads paper, which Linda Geiser can probably better cite right now off the top of my head, but in that paper, there were divided east and west models. So effectively, if you recall my map, which I could pull back up, there's a fair sampling gap in the center of the country where forest lands were not sampled for lichens. And then we sort of know that lichen responses, East Coast, West Coast, to get at the previous question also, are not uniform. So it helps improve, not just like improve model fit, but improve interpretability, biological interpretability to say an east versus a west model is adequate. So there's actually 4 models in that critical loads paper, east and west for nitrogen and sulfur. The idea of variability is really interesting to me. So that response surface is a mean air score. There's variation around the mean. So yeah, having a way to show sort of like 95% prediction intervals on that would be a better way to show that, to show the range of expected values.
Awesome, thanks. Any more questions of Rob?
I might add some nuance to the nitrogen sulfur question. In the West, the main reason we looked at nitrogen is because sulfur was very low at all the sites, really. Maybe a few sites around the west end of the Columbia River Gorge were a little elevated. But in the East, it's kind of hard to separate nitrogen and sulfur because there's so much of both. But where you can, the way I think of it, sulfur is, for one thing, in plants, and both nitrogen and sulfur are macronutrients. Nitrogen is often limiting, whereas sulfur usually isn't. In lichens, nitrogen is usually 10 times higher than sulfur. They just have a higher nitrogen requirement. And so the main effect of sulfur is more of a surrogate, it's an indicator of pH, which is very important for lichens, pH of the precipitation, pH of the substrates. So I feel like the nitrogen response is more related to nutrients, nutrition of the lichen, and the sulfur response is more related to the pH of the environment. So we tend to see this interaction between climate and nutrition more than an interaction between acidity and climate in the lichen work. But we haven't actually asked that question specifically, but that's the way that I think of it.
Okay, great, thank you. I think with that we are at time, so we can move on to the next speaker, who is myself. So I will time myself. We'll see how well I do moderating myself. And so I realized too, I just saw this in the PDF, there is a mistake in my title. I'm actually not going to be talking about monitoring with students. I'm not sure how that happened. But I am Niffer Wilkening. I'm a research ecologist. I'm in the branch of Air and Water Resources for the Natural Resource Program Center. We're part of headquarters for the National Wildlife Refuge System, and so we provide scientific support to all 570 national wildlife refuges that can be found throughout the nation. So this is really my first NADP meeting. I'm still learning a lot about NADP. But I've been working on climate change issues for almost 2 decades. I was trained as a climate change ecologist for both my master's and my PhD. And so, so Catherine and I sort of pulled in the rest of our branch, who are shown here as co-authors, to draw upon the expertise from both the water and the air side in our branch to kind of start thinking comprehensively about how climate change may influence air and water quality and what this might mean for fish and wildlife on our refuges. So we wanted to do sort of a demonstration project where we could provide some useful information to our managers that we could obtain relatively quickly and easily. And so this is our attempt to do that. And so a lot of the past research related to climate change and air or water quality is focused on humans or ecosystem services. This is starting to change. You can find more recent papers specifically talking about fish and wildlife, especially as we're seeing this increase in wildfires. But there's still not a lot of information that's out there specifically looking at fish and wildlife. And so this is by no means an exhaustive list, but this is just some of the ways that climate change may alter air and water quality, and that might matter for wildlife. And so this includes things like more heat waves resulting in a higher concentration of air pollution, more particulate matter from increased wildfires, larger amounts of airborne pollens and molds, and exacerbated water pollution due to warmer temperatures and more droughts. And so the ability of fish and wildlife on our refuges to tolerate elevated air and water temperatures and all of these climate change effects that they're already dealing with, we don't know how their ability may be compromised to do that with co-exposure to the increased pollutants. So we really don't know how most species will respond. So our mission in the National Wildlife Refuge System is specifically to conserve fish, wildlife, and their habitats. And so we manage a total of 835 million acres solely for wildlife conservation. We have a wildlife refuge in every state and territory in the nation, from the Caribbean to Alaska. And so we have this continental scale system of conservation. The majority of our refuges were created to protect waterfowl and migratory birds, so that's what we're still most widely known for today. But refuges also protect winter forage and birthing grounds for a lot of large mammals, nesting area for turtles and coastal species, American crocodiles. And then we also have a lot of critical habitat for threatened and endangered species on our wildlife refuges. And so we obviously didn't have enough time to do these sorts of assessments for all 570 refuges, so we picked 4 refuges to serve as case studies. We selected refuges in different ecoregions that are likely to experience a wide range of climate change effects to get a better understanding of how our refuge system may be affected overall. And so these are the 4 refuges we used as case studies. We have E.B. Forsyth in New Jersey, Okefenokee in Georgia, Bosque del Apache in New Mexico, and Lostwood in North Dakota. And so in the interest of time today, I'm going to show results from only 2, Bosque del Apache and Lostwood. And so we wanted to use these widely accessible, easy-to-use online tools to compile data for this effort. And so for climate change analyses, we use the Climate Toolbox. This is increasingly being cited in the published literature, and people are using it more and more across different fields. There's a lot of these online tools these days for running climate change analyses, and I actually think it's great. 10 years ago, we didn't have all these resources, so it's definitely become a lot easier to obtain and interpret both historical climate conditions and future climate projections. And then for the deposition data, we used our air quality trends, conditions and trends database. And so we don't really have a similar comprehensive database for water quality across refuges, so we use the information in this database as a proxy to kind of understand the potential effects on water quality too. And so the data key for interpretation is shown here. It categorizes trends as good, fair, and poor, and the overall trend can be improving, deteriorating, unchanging, or varied, and confidence level is also given in the database. And so for our first case study, Bosque del Apache National Wildlife Refuge, it's located at the northern edge of the Chihuahuan Desert ecoregion. It's about 60,000 acres and over 30,000 acres of designated wilderness. It was established in 1939 to provide a crucial stopover site for migrating waterfowl. The refuge is well known for the tens of thousands of cranes, geese, and ducks who winter there each year. We get a ton of visitors in the winter to see sandtail cranes. And so the historical wetlands have been drying out as water is diverted for agriculture and in urban areas. The refuge does have water rights, and so managers today are maintaining this habitat by moving water across the landscape, and this is certainly getting more difficult as this ecoregion is drying out. Okay, so what did we find out? So starting with some of the climate data. And so Climate Toolbox, I don't know if folks have ever played around with Climate Toolbox or Climate Mapper or some of these online tools, but there's a variety of metrics that you can use, everything from hydrology variables, things like snow water equivalent, to extreme other kinds of variables, heat indices, extreme fire danger days. So we started out using plain old temperature and precipitation for the first part, since we're looking at this at a large landscape level. So these are often the best metrics to start with for making comparisons at the scale. And so this is the spatial average of daily grid met data for maximum temperature, minimum temperature, and precipitation. And so the temperature plots are showing the annual means. You can see that both maximum and minimum temperatures have increased, and the precipitation plots are showing annual totals. And the annual total precipitation appears to be staying the same. You don't really see a positive or a negative trend, but then when you look at the annual max one-day precipitation, it appears that the values are increasing over the long term. And so this is interesting. You're going to see this again. But this annual max one-day precipitation, it's the maximum precipitation event from each year. So what this means is that the refuge is getting more of their precipitation in a single event rather than throughout the year. Or in other words, you can think of it as extreme precipitation events are increasing. And then for the future climate projections, we imported the refuge shapefile into Climate Toolbox to delineate the geographical area of interest, and then we used an ensemble average of 20 climate models and 2 emission scenarios, a high one and a low one, and this was downscaled to a 4-kilometer resolution. And so again, maximum and minimum temperature are projected to continue increasing, whereas this annual precipitation is not projected to change that much when you look at it this way. But when you start breaking this down and looking at different climate metrics, you start to see a slightly different picture. And so these box plots show annual soil moisture, which it considers precipitation levels and temperature. And so soil moisture is projected to decline under both emission scenarios. And so that makes sense when we think about the soil will be drying out since the temperatures are getting hotter, but the precipitation is staying the same. And so I picked this metric specifically because I know that moist soil units are an important component of maintaining habitat for this particular refuge. And so to look at the data from the Air Conditions and Trends database, so this is generated from the NADP monitor located at Bosque del Apache National Wildlife Refuge. It's this fair condition for ecosystem health. It's based on the 5-year average estimated wet nitrogen deposition of 1 kilograms per hectare per year. No trend information is available because there are not sufficient on-site monitoring data, and the degree of confidence is medium. And so the bottom figure shows the 3-year average of estimated total nitrogen deposition in kilograms per hectare per year. And so total nitrogen is declining. I think you can see that. And so that indicates an improving trend. And now to look at visibility. Visibility at Bosque is measured using an improved monitor. And the visibility benchmark is fair based on the 5-year average estimated visibility. The 10-year trend in visibility remained relatively unchanged on both the 20% clearest and haziest days. So this resulted in an overall unchanged visibility trend. And the degree of confidence at Bosque is high because there's, again, an in-refuge or nearby representative monitor. And then for the last one, looking at ozone. So for human health, ground level ozone concentration are fair at Bosque. This comes from the 5-year average estimated ozone concentration compared to the NPS ozone benchmark for human health. And then for vegetative health, ground level ozone concentrations are poor. And this is also based on, in comparison to one of the NPS benchmarks. And so for both of these, no trend information is available, and the degree of confidence is medium because estimates are based on data from more distant ozone monitors. And so to sort of summarize this and put this all together and think about what this information, what we can tell our managers based on this information. So the projected higher temperatures may increase ozone levels, as Emmy was talking about, so that was great to follow that, which is already poor for vegetative health, so that might be something that managers might need to be thinking about in the future. With the precipitation projected to be the same, but these much higher temperatures, it's likely that there's going to be much drier conditions and less surface water. And so this could result in reduced air quality. There's already increased wildfires in this part of New Mexico where Bosque occurs. And there can also be a higher concentration of pesticides or fertilizers in the water sources. They're already having a lot of issues with water availability on the refuge, and that's just going to get worse in the future. And so for our second refuge case study, we're heading farther north and east to Lostwood National Wildlife Refuge. This is in the Great Plains ecoregion. This refuge was established in 1935. It contains the largest public-owned tracts of northern mixed grass prairie left in existence. and it's a vital breeding and nesting ground for migratory birds. It also provides essential habitat for rare grassland bird species. So this refuge, the entire refuge, has been designated a globally important bird area. So just exactly like what we did for Bosque, this is the spatial average of daily grid met data for maximum temperature, minimum temperature, and precipitation. So the temperature plots are showing the annual means. So both maximum and minimum temperature have increased, but not quite as much at Bosque. Precipitation plots are, again, its annual totals. There doesn't appear to be much of a trend, but when you look at that annual maximum one-day precipitation, it is increasing, especially over the last 10 years, and the magnitude of this increase that you can see is greater than we saw in Bosque. So the annual maximum one-day precipitation went from 1 inch in 1980 to almost 4 inches in 2020. So that's quite an increase. And then for future climate projections, we again use the same thing, an ensemble average of 20 climate models, 2 emission scenarios, downscaled to this 4-kilometer resolution. Both maximum and minimum temperature are projected to continue increasing, although it's not as extreme at Bosque. It seems to be a lot cooler overall, and annual precipitation is not projected to change that much, although there may be this slight increase as it goes over time. And so again, looking at a different climate metric in Climate Toolbox, so these box plots show the length of growing season, and under both emission scenarios, it is projected to increase. And so this is what may be more important for lost-wood managers. There's a lot of problems with invasive plant species on the prairies there, and they're going to be battling invasive plant species. They could be battling these species more in the future, since many invasive plants respond positively to longer growing seasons and could outcompete native species. And so looking again at the same data from the Air Conditions and Trends database, wet nitrogen deposition levels create fair condition for ecosystem health at Lostwood National Wildlife Refuge. There's no trend information is available, and the degree of confidence is medium. And then the 3-year average of estimated total nitrogen deposition is higher than what we saw for Bosque, but again, it's declining, so it indicates an improving trend. Visibility is fair at Lostwood based on the 5-year average estimated visibility, and the 10-year trend in visibility improved on the 20% clearest days and remained relatively unchanged on the 20% haziest days. So there was an overall unchanged visibility trend. The degree of confidence is high because there is an in-refuge or nearby representative monitor. And so finally, the last one of these, looking at ground level ozone concentrations, which are fair for human health but good for vegetative health at Lostwood National Wildlife Refuge. And for both of these, the 10-year trend remained relatively unchanged and the degree of confidence is high. And so putting this all together for this refuge, for Lostwood National Wildlife Refuge, and so some of the things that managers may be thinking about is facing an increase in existing invasive plant species as growing conditions continue to become more favorable, and increases in precipitation and more extreme precipitation events may negatively impact a lot of the important birds there. And so just in conclusion, what we tried to do here is provide a blueprint for refuge managers to start thinking about how climate change may be changing things on the refuge and what this means for fish, wildlife, and their habitats. And so these kinds of data can also be used to inform NEPA and permitting actions and to guide climate change adaptation strategies and mitigation efforts. And that is all we have. I think I have time for questions. Mike, were you timing me? Okay. Cool, thank you.
I have one that we can start with. I know we've talked about this, but like as you're reaching out to refuges and identifying some of these changes, do each of the refuges have like a data on the species that they're interested in or the distribution of them so that we can connect them to some of the greater analyses like that Emmy was talking about and that Claude is doing?
Yeah, so that's a great question, and we talk about that a lot in the refuge system. And the short answer is it's hit or miss. Some refuges and some species, we have great comprehensive data, and so you could do some analyses where you're relating, survival of a particular species, or you're relating occupancy or things like that. But then in other cases, we have nothing, or we have, somebody went out like once a year at different times of the year for 5 years in a row. It's not standardized and it's comprehensive. So yes, it's a great question. I think that is one of the next steps that we're thinking about as well, because ultimately we want to be able to answer those questions for our managers in terms of like, well, how do I change my management for this species based on this information? So.
Very, very interesting stuff. You and the previous speaker are illustrating and showing, documenting negative effects of a change in climate on the ecosystem. But I wonder, are there any positive effects? Could the changing of climate actually enhance the protection of these ecosystems and species?
Yeah, so that's another great question and a conversation we have all the time in the climate change world, this idea that there are winners and losers, right? And I actually have moved away over the last couple of years from using that terminology, especially in reference to certain species. But the answer is yes, right? I mean, there are some of these changes. And so I think for this particular project, what I think about is some of those changes in precipitation levels. So in some of these areas where we're going to have more flooding and more precipitation, and there's a lot of places in the United States where that is projected to occur, it could actually improve water quality, right? Which is better for fish and amphibians and all the other aquatic critters inhabiting those areas. So that's one example, and I'm sure there are others.
Thank you.
Okay, great. I don't see any more questions, so I will attempt to get us back on schedule here. There we go. Okay, great. Our next speaker is Beck Dalton.
Great, thank you. I'm excited to talk to you today about a meta-analysis that I've been working on, looking at how soil microbial communities respond to nitrogen addition experiments under climate change. And I've been working on this project with a colleague, Tara Griever. And just before I start, I'd just like to give the disclaimer that the views expressed in this presentation are mine and not those of the EPA necessarily. Okay, so as we all know, nitrogen is an essential element for growth and development of organisms. However, we know over the past century that there's been large increases in nitrogen deposition over pre-industrial levels due to vehicles, biogenic sources, industry, and utilities. These can have detrimental ecological effects. They could include changes in ecosystem structure and function, such as acidification of soil and surface waters, and changes in species composition. And we know that nitrogen deposition in the US has been steadily, remaining the same due to some protections, but we're seeing a different type of nitrogen form becoming more important. So this is an image of oxidized nitrogen deposition on the left from 2000 to 2002, and reduced nitrogen deposition on the right. And what you can see compared to 2016 to 2018 is that we're seeing less oxidized nitrogen and greater reduced nitrogen in some parts of the country. Now, nitrogen deposition isn't happening alone. We're seeing precipitation and temperature changes happening at the same time. And so these are maps from NOAA looking at annual precipitation change from 1991 to 2020, minus 1981 to 2010. So we're seeing percent change. And what I'd like you to take away is that across the country, we're seeing drier conditions in some parts of the US, wetter conditions in others, as Niffer just mentioned, and generally warmer temperatures. And so this begs the question, does climate affect the sensitivity of microbial communities to nitrogen deposition? Okay, and so we can think about ways in which microbes are affected by nitrogen deposition in a forest. Nitrogen could have a positive effect on microbial growth or a negative effect, which I'll describe in these like pink and blue boxes. So nitrogen deposition could actually lead to a decrease in soil pH, which could cause an increase of leaching of magnesium and calcium ions. It can also lead to a reduction in enzyme activity, a reduction in below ground net primary production, and carbon, which could cause a loss of microbial growth. However, we may see some fertilizing effects of nitrogen where we see an increase in above ground net primary productivity, litter quality, and CNN, which we could see greater microbial growth. And there are ways in which climate can affect this response. So this is a lot, and I'm not going to ask you to read it all, but let's say as an example, we see an increase in precipitation. That could further decrease soil pH, causing the increase in the leaching of ions and a further decrease in microbial growth. However, increases in temperature, we could actually see greater above ground net primary productivity, causing increases in microbial growth. So we have a lot of different ways in which climate can affect sensitivity to nitrogen. Okay, so for this particular project, we did a meta-analysis examining how different nitrogen addition experiments in combination with increasing temperature, increasing precipitation, decreasing precipitation, and CO2 as well affect microbes. And I was trained as a field ecologist, and I'm always really interested in how people manipulate these types of climate effects in the field. So just to give you a brief overview, people use open-top chambers or infrared heaters or buried heating cables to manipulate temperature. And these are some of the most common. Either reducing or increasing precipitation can be done using these rainout shelters. So basically, you collect the rain that falls, and then sometimes they'll transfer it to other plots if they have both a drought treatment and an increased precipitation treatment. And finally, CO2 is often manipulated using open-top chambers or carbon dioxide delivery tubes. Okay, so getting to the methods of this, we did a literature search through Web of Science and PubMed. We ended up with about 9,000 references. And we were looking for nitrogen addition experiments that also manipulated some type of climate change factor from 1970 to 2023. And I would like to recognize Jenna Strawbridge of the EPA library who really helped lead this part of the project. I then used a program to screen abstracts, so I didn't read all 9,000 papers, but it got me down to about 185 references. And in this histogram I'm showing you, this is from 1996 to 2023, these are the number of publications that are included in our final study set over time. And we're not actually done extracting 2023 yet, so there's more data coming. And we ended up with about 70 references so far. Okay. And most of the papers that we found tend to increase precipitation. It's a very easy thing and cheap to manipulate, either through hand watering or those chambers. CO2, we find fewer papers. Okay, so far I've extracted about 4,000 data points from these references, and we're still not done. And we have a host of different responses, and the ones I'll be talking about today are bacterial biomass, fungal biomass, and the ratio of fungal and bacterial biomass, which is a good indicator of how much carbon these systems can potentially sequester. And as I mentioned, we're still extracting data. I didn't extract any soil characteristic information, which was a mistake. And we're still extracting references from 2023, and Emma Leith joined our project, who will be taking this on. Okay, so what I did was I calculated the log response ratio, which is basically the difference in the treatment effect over that of the control. And I'm working on using a Bayesian approach that basically allows me to look at multivariate responses, because a lot of these studies report more than one type of community response, and a lot of them are related to one another. It also helps us account for incompletely reported site or study level data. You would be surprised to know how many people don't report very common metrics, including sample size, and multiple treatment factors. Okay, so now to finally walk through some data, we found that experimental climate actually weakly influences microbial community responses to nitrogen deposition. So I'm going to walk through an example of the data that I'll show you, and then I'll show you real data. So on the x-axis is the log transformed ratio of means, which is basically the difference between the treatment and the control. And anything to the left of this dotted line at the zero means that there's a negative effect of the treatment relative to the control, and anything to the right indicates a positive effect. And I have the temperature, supplemental precip, and reduced precipitation experiments on the y-axis. So let's take, for example, in a nitrogen addition experiment where temperature is also manipulated, but these are just the nitrogen ones, we may see a decline in nitrogen, in bacterial biomass. However, temperature may actually increase bacterial biomass, as I pointed out in our previous example. And so the combination of these treatments, we may actually see no significant effect if they somehow cancel each other out. And so we can look at this for fungal biomass and the ratio. Okay. So now to show you some real data, which is messier. We see a lot of variation across all of these different responses and all of these different types of manipulations. And what I would like to spend more time on this talk is looking at how these supplemental precip experiments work. It's some of the most data. So what factors actually contribute to this variation that we're seeing, where we see some systems are showing increases in fungal biomass, for example, while other systems are showing negative effects on fungal biomass. Okay, so we know that there are additional factors affecting this response to N, not just climate alone. So there's been a number of meta-analyses published looking at different ecosystems and climate effects of this response, and we see a lot of differences depending on like the soil characteristics and the mean annual temperature and precipitation. We also know that the form of nitrogen used in these experiments does affect the microbial response. And so what I'll show you is a similar graph here, where this is just for the supplemental precipitation experiments. They either use urea, ammonium nitrate, or multiple types of fertilizer. And just showing you again, for bacterial biomass, fungal biomass, and FB ratio, we do tend to see a lot of variation across the different types of fertilizers. So there's still a lot of variation when using ammonium nitrate. And again, this is not, people also use different types of nitrate fertilizers, but this is what we have for these particular endpoints. Okay. We also know that the treatment magnitude and duration does affect these responses. So how much nitrogen is added and how long these experiments take place. Some of them take place over 9 to 10 years, while others are just a few months. And so we know that study duration does influence the fungal response to nitrogen and supplemental precip. So again, this is a similar kind of figure, but this is the effect of study duration on the response. And what I'd like you to take away is that we tend to see a negative relationship between study duration and fungal biomass response. And I'll explain this in the next slide. So if we look at treatment duration over time in years on the x-axis, and the log response of the fungal biomass in these nitrogen and supplemental precip results, we see a slightly negative relationship. And what we're seeing is that there's more variation in sensitivity to nitrogen deposition in supplemental precip when experimental duration is shorter. And there's so many different things that could be going on here. One of them is that fungal communities have typically lower nitrogen demands, and so we might be seeing this spike in fungal biomass at first, and then it becomes kind of a detrimental effect over time. The other thing is that the lifespan of fungi and bacteria are slightly different. We know that bacteria in E. coli lifespan is hours. In the soil, it can last up to days, but fungi typically last 25 days to 1 year, so maybe the response at which we're seeing fungal communities take a little bit longer to respond than bacterial communities. And finally, we know that soil characteristics can be really important, so soil pH, for instance, as I showed in this earlier slide. These data are not complete. This is only a handful of studies that we've been able to extract up to this point, but we do see some variation in soil pH across experimental treatments. So on the x-axis, I'm showing you the control, the nitrogen, the supplemental precip, and the supplemental precip by nitrogen experiments, and soil pH is on the y-axis. And we see some differences. I think it's mostly due to the difference in variation. So that's something that we'll have to be incorporating into our models. So just to wrap it up, in conclusion, we're finding that for some responses, microbial communities are pretty weakly sensitive, or there's a lot of variation to the effects of nitrogen and climate. We tend to see negative fungal biomass responses to nitrogen, and it's influenced by study duration and soil characteristics. And then I'm going to be continue working on this analysis to understand how the different types of nitrogen, study duration, and ecosystem affects these responses, and then also including repeated time series. So a lot of these studies will measure these, take snapshots of the microbial communities over many, many years and trying to put that into our models. And with that, I would be happy to take any questions.
Thanks, Beck. So when we tried to look at soil microbial responses to nitrogen 10 years ago, there was so much variation in what people were doing that it was hard to summarize. With the papers that are available now, are there enough common methods that you'll be able to look a little bit closer at the fungal community and say functionally or even phylogenetically, like who the winners and the losers are?
Yeah, that's a great question. So I would say over the last 10 years, there's been an increase in the number of papers that are reporting common metrics. A lot of folks will use very similar methods like phospholipid fatty acid analysis to kind of look at these fungal communities. And a lot of people are also reporting like relative abundances of dominant phyla. So we could start to see who are these, like which groups are actually increasing maybe at the expense of other groups. So that's a great question.
So given that a lot of these studies started in 1996, which was past peak nitrogen deposition, are you taking into account previous deposition that occurred and modification to condition, like initial conditions for each of these studies?
Yeah, that's a great question. So a handful of sites do report like what the average deposition is at their sites, and in the US we can pretty easily get that data from NADP data. It's harder for us, you know, it depends on what the sites are reporting, but that is something that we're including in our model is like what the average deposition is at these sites during that time period. So, great. I think that's it. Just please report your sample sizes and your error bars in all of your papers, or you'll be receiving emails from people like me. So, cool. Great. Oh.
Hi, this is really great talk and kind of addresses something that I've been thinking about the whole time, that, you know, I see a lot of studies that are just one variable, two variables, and this is really multivariate is what you're getting at. So I wonder if you could talk about like the state of the science for this to inform models. It seems to me like maybe biochemistry is so complex that they're not really process-based models right now, they're all empirically based, because it seems like what you're doing would really inform a lot of, you know, empirical modeling. And I wonder if you could, like, you know, talk about that.
That's a good question. I think I would have to think about that a little bit more, but maybe looking at like exposure response of these groups over large scales, we can start to see what some of these relationships are for certain phyla, and maybe inform some of the models that you're talking about. But sorry for the non-answer, but maybe I'd love to chat with you after. Yeah. Okay, thank you.
Okay, great. Our next speaker is Todd McDonald.
Okay, thanks NIFR and thanks NADP for having me. My name is Todd McDonald. I'm with ENS Environmental Chemistry. We're located in Corvallis, Oregon, and we partnered with Mike Bell and Emmy Felker-Quinn on this work. So just a quick overview. The project that we'll present today is related to understory vegetation response modeling across 8 national parks. One of the main focuses of the project was to evaluate critical loads of nitrogen deposition for understory plant species in these parks under current climate conditions. And some of this I showed last year, so it's a little bit of a review. with some new material on the response to climate change and also critical loads of N deposition in the context of future climate. And so we have completed 8 parks so far, and we have others in progress. So just why are we studying the understory vegetation? Understory serves a variety of ecological functions. There's great diversity in the understory. Some of what goes on in the understory can control what eventually occurs in the overstory. The species provide forage for a variety of wildlife. There's a variety of ecosystem services as well provided by the understory vegetation. So we used what we call the PROPS models to evaluate vegetation response. And the PROP stands for the probability of occurrence of plant species. So it's effectively a logistic regression model. We have 9 variables in the model. We have aspects of climate, N deposition, soil characteristics, light availability. So mostly the primary drivers of plant occurrence. And we have presence-absence data from complete vegetation surveys from around the country. And so here's just an example of a one-dimensional response. This is sort of a hypothetical response to nitrogen deposition on the x-axis, showing the presence and absence of the species. So this species would prefer a nitrogen deposition level around 5 kilogram of N per hectare per year. So we have more than 1,500 species characterized with these models. And they were developed with Chris Clark at US EPA in conjunction with several researchers at Wageningen University in the Netherlands, which is where this method effectively began. So we did a number of site-level studies with the models just to kind of get a feel for how they were working and do some testing, and we made a second version during that process. But our first regional application of the models was to Great Smoky Mountains National Park. The Park Service has these great vegetation mapping data sets that they've been able to develop from extensive vegetation surveys throughout the parks. And so what we did was we used those plot-level vegetation surveys to identify characteristic species that are associated with these vegetation map classes. And we did that by basically looking at those survey data and identifying species that were commonly occurring. So this is species that you'd expect to find if you're out in one of these vegetation map classes. And so then we used the PROPS models to estimate changes in occurrence probability for these characteristic species. And we did that under a base climate scenario of ambient climate conditions. And we also evaluated future scenarios of climate change. We're mostly focused on looking at warming at this point. So we calculated critical loads to protect the present day maximum occurrence probability. And this is an important concept that will come up later when we're talking about the critical loads, especially for future conditions. We generated those critical loads again under current and future climate scenarios. So again, here are those 8 parks. We're looking at Yosemite, Joshua Tree Glacier, Grand Teton, Rocky Mountain, Sleeping Bear Dunes, and Acadia National Park. And these are the climate scenarios that we've been evaluating: a 0.5 degree, 1 and 1.5 degree C increase in air temperature relative to the ambient condition. So the 8 parks are completed, and we have 165 that are currently in progress. So the results here will be for the 8 parks. So these are the critical loads of nitrogen deposition under the base climate scenario. So these box and whisker plots, each individual box and whisker plot represents the critical load for a given map class for a given park. There's 3 different types for each park. We'll talk a little bit more about how we develop these. There's different types of critical loads that we generated for each park. And one is the minimum. We call that the minimum critical load, and that's protective of all species, all characteristic species that we modeled. We also present information based on the 10th percentile critical load. That's protective of 90% of the species in that map class. And then we also are presenting results based on the median response or the median critical load among species within a map class. And so when we're trying to protect all species, we have very low critical loads. You see that you almost can't even see some of those green bars for several of the parks because those critical loads are driven by species that have a decreasing response to nitrogen deposition. So any level of increase in deposition is going to negatively affect the occurrence probability for that species and for a decreasing species. So when you're using a decreaser to represent your critical load for a MAP class, you have those very low critical loads. If you allow some damage to some species, your critical load increases. So we have increasing critical loads all the way up to this 50% mark. And we can choose any point on this distribution of critical loads across the species within a MAP class. These are just 3 examples. So we see across these 8 parks that most of these critical loads are below about 5 kilogram of N per hectare per year, somewhat regardless of which type of critical load we're looking at. I mean, the median for some parks does get up around that 10 kilogram mark, but for the most part, we're below or around 5. So this is a plot of the percent change in occurrence probability, so we're not looking at critical loads anymore. This is just the deviation in the maximum occurrence probability for the most sensitive species in a given MAP class for each park. So just to orient you to the plot, the 0 mark in the center, kind of similar to what Beck was showing with her microbial responses. And then so anything to the left of that line is a decrease in occurrence probability. And anything to the right is an increase. And since we're talking about the most sensitive species here, we're seeing decreases most often. So there are a few MAP classes there in Yosemite that their most sensitive species is actually an increaser. So yes, some of these parks we see increasing detrimental effects with increasing temperature to the point where some parks have their most sensitive species is no longer expected to occur under that more extreme warming scenario. When we look at the average of all species in a given MAP class, the results tend to be a little bit closer to 0. They do tend to have more, on average, across all characteristic species, tend to have a decreasing response. Although for some parks, we're seeing, like especially Grand Teton, we're seeing positive effects on average. And then Yosemite is kind of straddling that 0 mark. And just an example of how we can then map the results. So this is the same on the left, the most sensitive species for a MAP class. This is the change in the occurrence probability for that most sensitive species mapped just across some subjective classes that used for mapping. So we see the orange and red colors are decreases. There's a few little green specks in there where the most sensitive species in a MAP class is actually an increaser to some extent. And then like we saw on the previous slide for Grand Teton, there actually are a number of species that are favored by warming conditions. However, in those high elevation areas, we still have decreases even on average across all species. And this is just an example of what we can do to actually drill into the species level information to generate species lists by parks that are either more or less vulnerable to warming. So these are the species that are expected to have more than a 95% reduction in their chance of occurrence with 1.5 degree increase in future air temperature. It's a relatively limited number of species compared to the total number that were evaluated for a given park. But we can create these lists, and we can create this list for any level of reduction or increase as well. In some cases, if an invasive species is going to be increasing, that's something that a park may want to know about. So the title here says critical loads of future warming, but based on future warming, but here in the blue box is still, these are the critical loads, a summary of the critical loads under ambient climate conditions. So this is the same data that was on that box plot, the minimum 10th percentile and median critical loads, although then we're taking the median values of those distributions that were on that plot to really condense it down into one number that can be representative of the park. And so for Acadia, that median minimum critical load is still very low, and for the most part, other parks as well. And then see those increasing critical loads based on the 10th percentile, and then the median critical loads are higher. So then the next 3 segments of data are critical loads are based on the increasing temperatures. And this is where it gets a little complicated and where that maximum occurrence probability becomes important. We specified the critical load to the biological endpoint to be the maximum occurrence probability for a species under current climate conditions. And based on what we saw on the responses to climate change, what's happening is that the climate is expected to decrease the occurrence probability for, by and large, for enough species to disallow the reductions in nitrogen deposition to compensate for or offset those adverse effects from warming. So in those cases, we say that the critical load is not attainable. There is no level of deposition that can be reduced in order to achieve maximum occurrence probability under current climate. So this is all based on the definition of what a critical load is. If you're just going to determine the critical load based on maximum occurrence probability under the future climate, yes, we would have a critical load. But if we're trying to protect the present day occurrence probability, that's when we have a difficult time, in a lot of cases, being able to reduce nitrogen deposition to a level that can compensate for those adverse effects from warming. And this is very similar to what Rob Smith showed in that two-dimensional plot where he was showing those arrows moving around that data space. So in conclusion, many of the critical loads that we generated for these parks were either at or below 5 kilograms of N per hectare per year. Most of the increases, the projected increases in temperature tended to have adverse effects on plant species, particularly for those mountainous parks or mountainous areas of parks, and that reductions in nitrogen deposition and greenhouse gas emissions would be expected to preserve existing biodiversity in U.S. national parks based on these results. We'll take any questions.
Hey, Todd, thanks for that talk. I wonder if you have any thoughts about what's driving the negative impacts on air temperature on, the species, the sensitive species that you looked at. The reason I ask is, what we see in the Northeast with warming climate is herbivore browsing intensity tends to increase as winters become milder. In the Northeast, it's white-tailed deer, but it's other drivers in other parts of the country. Do you think that could be playing a role in those relationships?
I suppose that's possible. I mean, these are responses based off the data that we provided, and so what I feel like the biggest reason why we have those large decreases is because those are species that are basically on the edge of their climate niche, and so we just don't have. So they don't tend to occur in those. In the future climate scenario, and so there's a strong decrease when you get out to that level. What physiologically is driving it? I think it's probably more of a physiological response of the plant. But I suppose grazing could also play into it as well. But I think that would have to also statistically kind of coincide with the data in a way that kind of masks, overlaps with that climate response.
Thank you very much. I want to go back to the box plots that you had for the average total for the species. I noticed that Yellowstone and don't remember, Grand Teton seem to have the largest, at least whiskers of the box plot, and I wonder if you know why that might be. I noticed they both have the 2 highest map classes, but they're not the highest species number that you looked at, so is that maybe diversity? Is it the location of those parks? Is it the number of map classes that you used? Why do you think those have the largest extent that they're covering?
So for Yosemite?
Yosemite and Grand Teton both seem to have the largest whiskers.
Mm-hmm.
Their box plots extend further than all the others.
My thinking on that is that not only there's a fair number of different types of map classes in these parks, and a fair number of species that we modeled, and that these parks have a wide variety of ecosystems, and so we're just having lots of different habitats, and then we have lots of different responses associated with those habitats.
Thank you.
That's where I chalk it up to.
Since you're on this, can you go back like 2 slides? Yeah, I think that's what I wanted. I was thinking that, do you think there's value in creating like a weighted single critical load value like of these based on the areas of the different map classes? So if a park wanted a critical load value, it's like, well, if 50% of it is that point on the end, and that's going to shift the impact of each of these critical loads. Like, have you looked at that, or do you think there's value in doing something
We
like that?
haven't looked at that. I do think there's value to that. I mean, I think that's probably more of a question for the park managers. I mean, a rare species that may occur in just a small number of habitats or one habitat that occurs over a small area still may be, important to the park, even if it's only found in a few locations. But yeah, I mean, I think if you're trying to look at just kind of maximum impact, looking at it and weighting it based aerially, yeah, that would make sense. Yeah, we haven't done it, but we could certainly do that.
One thing I've been thinking about is when we did the lichen work, we saw a non-linear response of our climate or the lichen communities to climate change. So there's some places where the amount of community change was really in line with the amount of temperature change, and in other places there was very little change. And my wondering, which I would like to ask you if you've thought about this for the herbs, is could that be related to the initial starting climate? So, for example, the places we saw the least change were also places that already had the most extreme climates, where the climate can switch from very cold to warm in a single day, and as opposed to places that have more moderated climates where the vegetation is not adapted to extremes. And so you already have this kind of climate-adapted flora in these more extreme environments, so you would expect to see less impact of change. I wonder if you've thought about that or have any ideas about that for the herbs.
I mean, I suppose that's possible, yeah. I mean, I think you do have this issue of being, climate adapted, and whether or not that, can make you more or less resilient, to climate effects. How that plays out in these data, we really haven't dug into that. But yeah, I think part of this, getting in, there's so many species and so much information that, right now we're at a fairly high level on it. It's kind of summarize, trying to produce information that the parks can use because they want to have it kind of aggregated. But to drill into specific species, especially species that are studied well, and people, species that have information, have a lot of, there's a lot of knowledge around that species in terms of its niche and its flexibility in terms of adaptation. I think that could really help to dig into some of these questions. But
Well, my point is more by park to park. I mean, all species would have to be adapted to extreme climate changes in some parks that are more continental, for example, as compared to sites that are right along the coast, where it's more moderated all year. The entire community does not experience extremes, whereas in these other places they do. So what I'm suggesting is, I wonder if the whole community would be more or less sensitive to climate change depending on what climate, the current climate ranges are. Mm-hmm. Yeah, that's what I'm saying.
Yeah, that makes sense. Yep.
So just something to look at.
Mm-hmm.
Todd, I just had a question about the scope of the analysis. What fraction of the species do you think you're including of the species that are present? Like, is it your perception that you're including all of them, or is it, I know it's hard to get all of the herbaceous species at a location.
So you mean what fraction of the total species count in a park are we modeling?
Yeah.
Yeah, I mean, that's really variable. We have there's a lot of different map classes in these parks. And so you'd have to look at the map class level. But what we did was we said, if a map class has, say, 4 different vegetation surveys, plot locations on it, it would need to be found at least 3 of those locations. So we had, at least one of those locations. we had a 25% occurrence cutoff. So you needed to be found at least 25% of the vegetation plots that occurred in a map class. And so based on that criteria, we cut off species that were only found, so let's say there was 10 vegetation surveys and it was only found on one, that species wasn't included. It was not considered characteristic of the vegetation type. I mean, we're probably coming out somewhere around half of the species, I'd say. But that's, like I said, it's very variable by map class depending on what resulted from those vegetation surveys and that 25% cutoff.
So next, we are going to have a panel discussion moderated by Linda. Oh, and that's my timer. So that's great. We're right on time. And we are going to be talking about climate change, conservation, and critical loads. And so we need Emmy back up here, and who am I missing?
We've got you and then Rob on the computer.
And Rob online. Okay.
Thanks.
Sure.
Great. So I think it's pretty exciting where we are in terms of integrating climate into the critical loads and effects work. And I just wanted to kind of take a little bit of time to think about sort of where we're going in the future and think about how we can link together some of the things that we've heard today. So I guess I have, and I'll just ask a couple of questions to start it off, but I invite, it's supposed to be a discussion, so if people have follow-up questions, please ask the people here or Rob also. So, but I guess one of the things that I'm wondering is what you see as the biggest gaps for each of you in your work, and because I'm thinking about, priorities for where we're going, and then also thinking about linking your work together so that when we think about, we tend to, although this work is much more synthetic than some of the work that we were doing, say, 10 or 15 years ago, but thinking about it from the resource manager needs, a person is going to be interested in understanding how all of these different impacts are going to act together. So I'm just wondering if you see opportunities for linking together some of these different pieces of work. So I guess, why don't we just go from one side to the other, and then we'll get Rob at the end.
Okay, I think this is on now. So I can get us started. So that's an interesting question because I think this effort that Catherine and I embarked on and the rest of our branch addressed some of that. I mean, I think, again, so I'm coming at this from the lens of fish and wildlife, always, right? That's what I'm thinking about. And that doesn't mean that vegetation's not important for fish and wildlife, because it really is. But really thinking about climate change and, this nexus with behavior and health and physiology and all those aspects related to fish and wildlife, I think that is probably one of the biggest information gaps. I think we have, there's quite a bit of literature out there, and there's information out there about vegetation communities and thinking about climate change and what these communities might look like in the future, thinking about air quality. But there's really nothing for wildlife. And so, again, especially since we know some of these things are going to change breeding behavior, some of these things will change recruitment, some of these things may change survival, right? And we really don't have any information about what that looks like for a lot of fish and wildlife species. So,
And then sort of from the soils and waters and plant side, for us, one of the biggest barriers is understanding sort of where we need to be going with the synthesis. It's been interesting to take the automated reports of critical loads to managers and get really different answers from them on what they absolutely need to have. Smoky Mountains wants red spruce prioritized and the waterways that are still affected by acid deposition prioritized. When we took critical load synthesis to colleagues at Bureau of Land Management, they wanted a single value for the abundant species, right? I think we have so much great data now, and synthesis and deciding what it means is going to be, I think, I was hoping for a clear answer from each analysis, right? But there's still decisions to be made.
I'm going to just jump in before Todd, and I was, interested in some of the climate tools that you showed, Niffer, and I wonder about, as we move forward with the temperature and precip. I'm trying to find Justin, but I can't see. Oh, there he is. Okay. With the tree analysis of using some of those kinds of tools, because I think there's the analysis piece, and then there's also the communication presentation, kind of giving a clear schematic and presentation of what the impacts are, what the climate changes are, and then what the ecological impacts are. So that's, I think, something we might think about. Todd?
Sure, yeah. In terms of gaps, I mean, I feel very specifically soil chemistry, I feel like we're oftentimes, frequently looking for soil chemistry or just not able to use aspects of soil chemistry in these spatial models because they're just not available in enough places. So this is acid-base chemistry, and then also some estimate of nitrogen accumulation or just sort of nitrogen availability in the soils. So I think that that would be great in order to have some. It would need to be an extensive network to really be able to do the kind of scale modeling that we're doing now with these statistical models. But perhaps maybe we could expand process level modeling in those locations, and we may not have enough to spin up a very robust statistical model, space for time substitution type model. But maybe we could get back into some of the process-based modeling, and I think in those process models, you can interact these effects of temperature. Say you're looking at tree growth. I mean, you're mechanistically modeling those effects, and you can validate that and calibrate parameters when you're working at that site level. And if we had more sites to work at, we'd be able to characterize more ecosystems and perhaps maybe extrapolate that with a statistical technique after the fact. So I think soil chemistry would be great to have. And then in terms of linkages, I mean, taking this information, taking what the effects on lichens, the effects on understory plants, and being able to translate that into effects on wildlife species would be fantastic. I feel like we can find at least a few areas where we might be able to trace some effect on vegetation or lichen to a change in abundance in a certain wildlife species, or perhaps nesting habitat for bird species. Maybe it's not possible, but I would think that there would be at least one location that we could do this as a test case, and perhaps expand that. Use that as a way to say, okay, well, what would we actually need to do in order to be able to make those linkages? Yeah, I think that would be really interesting.
And I think there are some, I mean, I know in the Forest Service and probably in other agencies, identification of, say, rare or threatened species and the kind of next ecosystem component upstream that they're depending on. That could be a way of trying to connect that.
Yeah, and I've personally done some of those analyses. So yeah, you can look at survival, right, or occupancy, or some of those metrics, and relate those to both vegetation characteristics and how climate is ultimately driving both of those. I mean, they're complicated and they're time-consuming, and they often require a lot of data, right? And for a lot of wildlife species, we just don't have those, especially survival data, right? I mean, those are really hard data to get for wildlife. So, but yeah, I agree 100%. I think we're at that point, where we can take that next step and start trying to prioritize doing that more, because that's really what's needed. So.
And is Rob still on? I don't see him. Okay. Sure, Linda Geiser. I was thinking about calling on you anyway. So just thinking about gaps and kind of next steps, like what are the gaps facing your analysis, and also where do you see linkages potentially between other components that people have talked about?
Right. Well, I think the initial results from the 20-year data set that we looked at for lichens really interesting, mainly because of what I was saying to Todd, that we did definitely see effects of climate change, but it wasn't like pollution where you could mainly say, okay, the pollution is increasing, the community is shifting, and it's sort of a continuous thing, and it's happening one-to-one. You can predict the amount of nitrogen deposition change from this amount of community shift. With the climate response, it was, some places were changing a lot with a lot of climate change, but some places were not changing much with a lot of climate change. And so to me, that's like understanding why that's happening is really an interesting question. I have my hypothesis that it's related to, well, those areas are already extreme, have extreme climate. So they're already weeded out species that can't tolerate extremes. And so you're just putting a little bit of change on top of what is already a big annual, seasonal, even daily change that all the species in those places exist. But we didn't demonstrate that. We only saw that it wasn't an even response. So I would like to know, is this kind of thing also happening with the herbs and with trees? And could we look at those data sets together? Could we combine the lichen and herb and tree data set that's complete for some area and take a look over those 20 years? And now I'm knowing that you're doing veg plots for the Park Service. Hey, Forest Service has veg plots in that 20-year study area. Maybe we could use the ecology veg plots. And of course, the FIA data is available for this whole thing. So that's the kind of thing I would like to do next, is let's see if those same kinds of effects are uneven effects and why that's happening. I think that would be really helpful to know that for land managers, for example.
No, and it would be, I like the idea of trying to look across, and I think like a case study, like from one of the parks that Todd's already looking at or something like that, where you have lichen data to try to look across and see, see how far we can get with what we have and then what we really need in addition. That'd be kind of cool. And I think that would be great because we're running out of things to do. Yeah, so. Does anybody have any questions for the panel? Okay, I'm just going to ask one other thing, if there aren't any other questions out there, is, okay, go ahead.
Just a question to Todd for clarification. When you talk about a process-based model, you're not talking about like at the molecular level, like chlorophyll production. You're talking about like a more macro scale process, right?
Well, I think somewhere in the middle. I don't know if you're familiar with models like FORSAFE or PNET BGC, where you have a biogeochemical model that's driven with mechanistically, and that relates to a forest growth model. So they can, and then there's feedbacks. So, there's litter fall, and there's the whole ecosystem is modeled with equations based on response, based on literature values and equations developed from literature, and then parameterized and calibrated to observed conditions, and then be able to simulate historically and future conditions at a site rather than what we've been tending towards recently is more of a statistical kind of space for time substitution, statistical modeling framework. So if that helps.
Okay, so my final question is, let's say I'm a resource manager. How do I know what I should care about most? How do we, or to flip it around, how do we give people the information that they most need when there's a lot of different information?
I know you've talked about pristine parks and kind of the lichen communities there. Basically that if you're in a place with very low nitrogen, even a little bit of nitrogen added will cause a big change. Do you want to talk about that, kind of the places that have a strong legacy or of clean air versus industrial sites?
Yeah, I mean, I guess I think it's like, yeah, whatever you want. I just think, like, should I care most about lichens? Should I care most about the herbs? What should I care about? How do I know? How do I figure that out?
Yeah, I'll go ahead and take the first attempt, and then Linda, maybe that'll stimulate something for you too. Thinking, yeah. So we have related conversations a lot with our wildlife refuge managers, right? I mean, as most of you probably know, our refuge managers our refuges are extremely understaffed. We have all these refuges where we don't even have staff. We have a lot of refuges where, there may be a biologist, but there's no refuge manager, et cetera, et cetera. And so we are constantly faced with that, when we're out on a wildlife refuge in terms of our interactions with the managers. They're harried, they're running around, they're overworked, they don't have a lot of time, and they often do want to know, what do I need to care about here? You've got 10 minutes, to tell me this. And so, it's without the ecological context, right? Again, thinking about our National Wildlife Refuge System, it's going to be something different at each refuge. But I think what we strive to do is always think about it that way. In terms of first of all, being as brief and as relatable as possible, which I know we all do with our research, right? Try to translate it into something that, you can tell your refuge manager in 3 sentences, this is the most important thing and this is why you need to care about it. And that's not always easy to do, but yeah, I mean, it's a great conversation and, a good question, because I don't always have answers for that too, but I guess, what I'm saying is we are kind of wrestling with that too in the refuge system and trying to think about that, and being brief and relatable, is what we try to practice anyway. So I don't know.
Yeah.
Did that help? Are you ready now, Linda? Do you have?
Yeah.
Okay.
You have 2 minutes. You have 2 minutes, Linda Geyser.
Well, I would say, number one, that all of our agencies right now are already engaged in a climate response very actively, and that climate change is really, I mean, the elephant in the room for everything, and that within the climate change response, there's nothing that's more important than reducing greenhouse gas emissions. I mean, every single 1/10 of a degree that we can save in increase is going to reduce the impacts that we're going to be observing on the ecosystems that we manage. And so any opportunity that we have to reduce greenhouse gas emissions really needs to be a higher priority. And so within the Forest Service, for example, somebody's probably trying to give me a call. So within the Forest Service, for example, I mean, one of the ways that we could embrace that, right now we've got the wildfire crisis, and so we're dealing with reducing hazardous fuels, trying to reduce the impacts and make the ecosystems more resilient. But ultimately, if climate keeps changing, that can't be effective forever. And so can we rethink about how our forests, for example, instead of storing biomass in the trees themselves, can we actually use the trees to reduce carbon from the atmosphere and take actions where we keep wood products stored for long periods of time, for example? Anyway, I'm kind of wandering into this other realm, but the key message is greenhouse gas emissions need to be reduced in order for any of our other activities to be successful. So somehow those have to be prioritized with revisioning, like the things that we do, how can we do them to help achieve the slowing of climate change?
Great. Well, thanks everybody for your talks and for participating in this panel.
Well, before we close up, let's give them all a round of applause.